Good afternoon, students. Okay, hope you are going to clear. Hope my voice is clear for you. Uh, so before going for today's topic, I want to remind you once again the different types of OVA, based on which only you'll be able to understand what blastulation is and gastrulation is. Okay. Once again, to remind you again, once again, here, based on amount of yolk, eggs are of different types. One is said to be microlithalic, where the yolk is said to be very less. Mesothelistalic, yolk is said to be moderate, most of which is present in the vegetal pole and some of it in the animal pole. Even in the vegetal pole, if this is the equator, the yolk is present even above the equator in large amounts, and towards the animal pole, compared to the vegetal pole, it is in less amounts. Whereas in microlithal, like it is uniformly distributed, though it is less. Hence, these are also called as isolithal eggs, and these as moderately telolithal eggs. And coming to polylithal eggs, these eggs are said to be leaving only just a small space towards the animal pole. The entire vegetal pole, even above the equator, almost the entire animal pole is occupied by the yolk material, leaving a small space here. And egg is having a nucleus. The nucleus is present in yolk-free cytoplasm, which is called as active cytoplasm. Thus, here it is present there, and here it is present here. In addition to this, there is another one called as centrolithial egg, where the egg is said to be having yolk at the center and active cytoplasm towards the periphery, where the nucleus is located. And based on potency, developmental potency, eggs are again divided into two types: mosaic egg and regulative egg. And the development of the individual from this is called as mosaic development, and this as regulative development. In mosaic egg, what happens is, in a certain eggs, every portion, remember this, every portion of the egg is predetermined with respect to its potentiality or ability for further development. For example, if this is the egg, if you say this part is going to give rise to nervous system, if you remove this part, nervous system may not be able to develop. Thus, the part of the egg is said to be predetermined. If small portion of such an egg is removed, the embryo will be defective. That particular organ or system will not develop, because the removal of this portion results in the permanent loss from the egg, so that the remaining portion, after removal of the specific portion, it cannot make compensatory development to make good the lost part. That means whatever that is remaining after removing certain part, this cannot compensate the lost part. Such an egg in which the future developmental potentialities are predetermined are in the form of a mosaic. That means this portion is different, this portion is different, this portion likewise. Every portion is said to be different. Hence we call it as mosaic egg, and that development is called as mosaic development. Here, the destiny is predetermined, hence we can call it also as determinate egg. This is seen in annelids, mollusks, and ascidians. And second one is said to be regulative egg, and it is seen in vertebrates and in most of the invertebrates. The developmental potentialities are not predetermined. They are not predetermined. Here, they are predetermined, and here they are not predetermined. That means removing a small portion of the egg, that means one or two early blastomeres, is not going to affect the normal development. So this type of egg in which the future developmental potentialities are not predestined or predetermined is known as regulative egg, and the type of development is said to be regulative development. Here, as it is not predetermined, it is called as indeterminate egg. In case of echinoderms and vertebrates, we can see this type of egg. Okay, and coming to the, that is what the reason. For example, I would like to explain this topic. Uh, Human beings, after sperm fertilizes the ovum, it results in a zygote. Zygote divides to form two cells. If you separate these two cells, what happens? Each one develops into an individual. That means this egg is not this cell, the first cell, this is the second cell. This cell, in the case of mosaic development, let us say it is going to form half of the body and the other cell, the remaining half. So that one, if we remove one cell, the other half of the body will not develop. Whereas here, even if you remove one cell, that becomes what we call, again, that behaves as if it is a zygote. It is a totipotent cell, and it can develop directly into another individual because its fate is not determined. On the basis of shell, eggs are again of two types. Some are called as cladoic shells and the other called as non-cladoic shells. Cladoic eggs, as the name implies, cladoic, they contain a hard outermost shell. Though it is hard, it is permeable for gases. Otherwise, if oxygen will not go inside, there is no oxidation of food material, there is no release of energy, there is no breakdown of food material, there is no development at all. So, though it is hard, the shell is, it can permeate, it can allow the gases to permeate. Yolk, salts, and water are present in large amounts in these eggs. That is the reason that they are said to be exhibiting direct development. And this cladoic egg, why the shell is present? When a bird lays eggs, it lays on the land. If it is said to be not covered by shell, the scorching heat, to the scorching heat it is prone and the development may be impaired. If at all a shell is present, the embryonic development is not affected by the external temperature. Thus, it is a sort of terrestrial adaptation seen in insects, birds, reptiles, and prototherian mammals, which are egg-laying mammals, exemplified by platypus, jaglossus, tachyglossus. All these belong to prototheria. And the other one, where the shell is absent and the egg membranes are very soft, they are called as non-cladoic eggs, exemplified by all those that give birth to the young directly, or all those which lay eggs in water are said to be non-cladoic eggs. Okay, so with this brief introduction, that is, we already have discussed about the process of oogenesis. That's why I'm skipping it. Now let's see, after oogenesis, what happens is they develops a ovum or egg. We can call it as unfertilized egg or simply as egg or simply as ovum, whatever it may be. And this ovum is found to be having additional membranes around it. After its formation, it shows additional membranes around it. We can call them as primary, secondary, and tertiary egg membranes. Let us discuss these three different types of membranes. Coming to the primary egg membranes. These are formed by the ovary itself. Where they are formed? They are formed in between the egg plasma membrane and follicle cells. When you see, this is the germinal epithelium and this is a Graafian follicle. So the Graafian follicle has a number of follicular epithelial cells inside which there is an egg. So what the scientists say, the primary egg membranes develop between the egg plasma membrane, this blue color, and follicle cells, the green colored cells. So that means it will be like this between these two. And what secretes this, what we call primary egg membrane? Because it is present between the plasma membrane of the egg and the follicular epithelial cells or follicle cells, either follicle cells or the egg itself can secrete the primary egg membranes. In some case, both secrete the primary egg membranes. So that is it, as it is secreted by the follicle cells or the ovum or both. And these are of three types. In some it is called as vitelline membrane or vitelline membrane, and some it is called as zona radiata and also as jelly envelope. So questions may be asked, one with respect to which is going to secrete the primary egg membrane. Second one, where actually it is present. Third one, identify from the following the secondary egg membrane. So they will give these three, and another one, the secondary egg membrane, you have to identify it. Okay. If at all I identify uh, from the below options, primary egg membranes, these three are given, and for all of the above, you will answer all of the above. Next, coming to these three different types of membranes. First one is called as vitelline membrane. Vitelline membrane is said to be non-cellular and transparent layer. So this is the plasma membrane above which it should be present. So this becomes the vitelline membrane. So it lies over the plasma membrane and it is a mucoprotein layer made up of mucoprotein. And it is called as chorion in reptiles and it is called as zona pellucida in mammals. Whether it be chorion or zona pellucida, this is the one which recognizes the sperms of the same species. For example, in case of mammals, zona pellucida contains different proteins. We call them as ZP1, ZP2, ZP3. And ZP3 is the protein which is responsible for species-specific attachment of or adhesion of the spermatozoon with the ovary. Thus, this is what actually responsible for species recognition between the sperms and ova. And another function is that it holds up the dividing cells together during cleavage. So if this is the plasma membrane, this is a single cell. So this divides to form two cells and then four cells. And all these are said to be held within the vitelline membrane only. With each division, the size decreases. This is a large size cell. After that, two cells which are said to be half of this size are formed. And after that, two cells which are one-fourth of the size. Thus, four cells are said to be formed. And all these are held together by the vitelline membrane, which is present outside. And in case of certain organisms like sharks from elasmobranchs and some bony fishes, some amphibians and reptiles, the primary egg membrane present above the plasma membrane is called as zona radiata. So this is somewhat striated in appearance. It shows striated in appearance. And it is said to be representing the degraded microvilli of the growing oocyte. So the oocyte, we know plasma membrane is there. The plasma membrane in general can form microvilli in order to increase its surface area. Brush border, you know that during absorption you might have studied it. And these degraded microvilli, when they appear like a striated appearance, scientists call this as zona radiata. And the third one is jelly envelope. This is also present over the plasma membrane. So this is plasma membrane, this is jelly envelope, and this is much thicker when compared with the other ones, and it is present in echinoderms and some marine invertebrates. Thus, there are three kinds of primary egg membranes: white line membrane, zona radiata, and jelly envelope. So you have to remember these three names and also the examples. And coming to the primary egg membrane again, it is a small, what we call extra characteristic in certain organisms. The space between the egg plasma membrane and primary egg membrane. So this is the plasma membrane and this is the primary egg membrane. So we call this as vitelline membrane, so that this is said to be having a perivitelline space filled with perivitelline fluid. That is what actually happens. But in some eggs, there is a small opening found in the primary egg membranes of some eggs. In mollusks, it is situated at the vegetal pole. I have shown it in the animal pole, but actually it is in mollusks it is situated in the vegetal pole, whereas in echinoidea and pisces it is situated in the animal pole. So whether it is in animal pole or vegetal pole, this pore or opening present in the primary egg membrane allows the sperm entry. It allows the entry of the sperm. It facilitates the entry of the sperm. That is what the significance of this micropyle is. Okay. So this is the primary egg membrane, which is having a micropyle through which the sperm entry takes place. Next, coming to the secondary egg membrane. These are the membranes present outside the primary egg membranes. Okay. These are secreted by a layer of follicle cells that surround the oocyte. That means ovum cannot secrete it because ovum secreted primary egg membrane, so it is detached from it, around which there are a number of follicle cells, and now these follicle cells secrete a secondary egg membrane. So it is always secreted by a layer of follicle cells that surround the oocyte. In insects, ascidians and cyclostomes, this is called as chorion again. So there is a name called as chorion. The white line membrane is called as chorion in reptiles, and the same is repeated here also. The secondary egg membrane in insects, ascidians, and cyclostomes, which is in the form of a chitinous shell, is called as chorion. Another chorion term we come across with in the fetal membranes or extraembryonic membranes, where we have chorion, allantois, yolk sac, and amnion. Okay, so chorion is the term which is said to be coming off in different places. You have to remember those. And this secondary egg membranes are not found in case of amphibians, reptiles, birds, and mammals. So, because this is an exception, a question on this may be asked. Which of the following animals do not have a secondary egg membrane? So, you can remember it like this: all tetrapods. So, in tetrapods, starting from amphibia to mammals, the secondary egg membrane is said to be absent. And in case of mammals, though it is absent, some call the corona radiata as secondary egg membrane. But to be precise, it is not a true membrane at all. Membranes means the cells should be continuous, side by side with tight junctions, isn't it? If the cells are said to be somewhat detached from each other, like here, we cannot call it as a layer. So that is the reason why we are saying it secondary egg membrane is absent in mammals. But what happens is somebody have come. So in mammals, when a Graafian follicle ruptures and releases ovum, you know that, no? So this is ovary, this is Graafian follicle, and there is ovum. So the Graafian follicle ruptures and the ovum is released. As the ovum is released, around the ovum, what are they present? Follicular epithelial cells are present. So along with the ovum, certain follicle cells also are peeled off. They come along with it. They are present outside the primary egg membrane. And scientists call this as corona radiata. So corona radiata is considered in mammals as a secondary egg membrane, but actually it is not a true membrane. It is nothing but the follicle cells which are present outside the primary egg membrane that are carried along with the process of ovulation around the ovum. This is ovum. Coming to the third one, tertiary egg membranes. These are secreted by the cells of the oviduct as egg travels down the duct toward the cloaca. So these are secreted only. This is if this is the oviduct, the egg reaches the oviduct. So as it passes through until it reaches the cloaca from where it is delivered outside, here the egg is said to be invested by certain membranes. These are called as tertiary egg membranes. These are always secreted by the oviduct and they are of four types: albumin layer in some organisms, and in some it is called as jelly coat. In some, cell membranes and shell also constitute tertiary egg membranes. Coming to the albumin layer, it is secreted by the oviduct itself. The oviduct has shell glands, and these shell glands secrete the albumin layer. And it looks like having projecting certain horn-like structures, and in the form of a hard, thick membrane around it. And hence, we say that it is in the form of a horny capsule. It is drawn into long twisted horns. What actually these horns are responsible? When the eggs are released out, they are said to be entangled among the seaweeds so that they will remain there instead of going along with the water to somewhere else, to hold them into the seaweeds, among the seaweeds, so the sperm can encounter them. These rays are responsible for holding or entangling themselves among the seaweed plants. These are found in elasmobranchs like oviparous sharks and ways. The second one is jelly coat. In amphibians, three layers of albumin are deposited around the egg, and this is in the form of a jelly, and hence they call it as a jelly coat. What is the function of it? It holds the egg together in masses. That means large masses of eggs are said to be amphibian. In amphibian reproductive system, we know that when the male comes and onto the female and presses it, the female releases all the ova, and all the ova not separate. They are said to be crowded. Okay, so what makes them adhere into masses? The jelly-like substance, the albumin layer, three layers of albumin present around them, keeps them together in masses so that the eggs are protected from number one, infection, number two, it acts as insulation, and it makes them unappetizing to predators. So this jelly is not what we call palatable by the predators. So thus the eggs are most of the eggs are not eaten up by the predators. And coming to the final one, that is shell. In case of reptiles, birds, including chick, five tertiary membranes make up the envelopes of the egg. And all these include the egg contains three layers of albumin, two shell membranes, and a porous shell. All these constitute the shell which makes up the tertiary egg membrane of the reptiles and birds. And coming to the significance of egg membranes, the first thing is it offers protection from temperature, changing pH of the water, pollution, etc. Second one is it prevents polyspermy. Fertilization of the egg should take place by only one sperm. More than one sperm should not participate in fertilization. If one participates in fertilization with one ovum, it is called as monospermy. If many spermatozoa participate with ovum in fertilization, it is called as polyspermy, and the egg membranes prevent polyspermy. At the same time, they prevent fusion of different eggs coming together. Even they are in masses, the jelly coat separates each egg from the other, so that they prevent fusion of different female gametes. And to maintain normal cleavage of the egg following fertilization, where the different cells are said to be formed, as the number is increasing, all these are held together by the vitelline membrane, which is a egg membrane. And that is how the cleavage process goes within these egg membranes. Thus, normal cleavage of the egg following fertilization is maintained by the egg membranes. And coming to the next topic, that is fertilization. Fertilization is nothing but union of male and female gametes, where a haploid spermatozoon comes in contact with the haploid gamete. That is what we call it as fertilization. This is followed by the transfer of nucleus of sperm to the egg, after which the sperm falls off, and inside the egg both the nuclei fuse, a process called as amphimix. So fertilization is coming together of both the gametes, and amphimixis is the fusion of the nuclei of the gametes, so that a diploid zygote is said to be formed. And coming to the site of fertilization, where actually takes place. So based on the medium in which fertilization takes place, there are two types of fertilization. You know about it, external and internal. External fertilization requires a water medium. So in mostly where there is water medium, that means aquatic organisms can exhibit external fertilization, even amphibious organisms certain. And those where there is no water medium, all of them exhibit internal fertilization. So fertilization in aquatic medium outside the bodies of male and female parents is called as external fertilization. The aquatic medium may be seawater or freshwater. For example, here the amphibians show release ova and eggs, and the fertilization takes place inside the water. Coming to the internal fertilization, it is characteristic of terrestrial animals. It occurs in oviparous, that means egg-laying animals, viviparous, those animals that give birth to young ones, or ovoviviparous animals, that means animals producing the eggs that are hatched within the body of the mother. They produce the eggs, but those eggs are hatched within the mother and babies are delivered outside. And these types of organisms exhibit internal fertilization, where the male gametes are delivered internally into the body of the female. And here, the introduction of sperms requires a male copulatory organ. Okay, and coming to the mechanism of fertilization. For our understanding, scientists have divided the entire process of fertilization into 7 steps. One is spermatozoon has to reach the egg, that is approach of spermatozoon to the egg. After that, sperm has to adhere to the egg, that is what we call it as capacitation and sperm egg adhesion. And the third one, penetration of sperm into the egg membranes, through the egg membranes has to take place so that it comes in contact with the membrane of the plasma membrane of the egg. And after that, gametes fuse, we say so, and it prevents it. It is required as many millions of sperms reach the egg, it has to be prevented. The polyspermy has to be prevented. And once the polyspermy is prevented, in case of mammals, we know that during ovulation, it is not the ovum, but it is the secondary oocyte that is released. So, this is still the cell that is produced after first meiotic division. Now it has to complete, the secondary oocyte has to complete second meiotic division and form into ovum. And this is accomplished by metabolic activation of the egg followed by fusion of the genetic material, that is amphimixis or syngamy. And let's start with the first step, approach of spermatozoon. If it is zoon, it is singular. If it is zoa, spermatozoa, it is plural. So approach of spermatozoon to the egg. So whether it is external or internal, the sperm, because it is having a tail, which is nothing but flagellum. In the case of external fertilization, it shows swimming movements within the water. In case of internal fertilization, it shows swimming movements as it passes through the female reproductive tract along with the semen or the secretions secreted by the female reproductive tract. So in both the cases, it has to reach, for which the flagellum helps. And this is accomplished in different organisms in different ways. And in organisms, in many of the organisms, it is a chance encounter or random. Okay, so all are said to be, there are different types of species. For example, there is a frog, Rana pipiens, in the same pond, and there is Pipa americana in the same pond, and there are elasmobranchs, there are bony fishes, so many are there here within the same pond. So everything is said to be, if they are said to be involved in external fertilization, all the sperms and ova are released into the same pond. So here what has to be done? The sperm has to reach the egg of its species. So during that, it may encounter certain obstacles like submerged plants, floating plants, other animals, those that can that are predating on these sperms. So it has to encounter and reach the destination, that is ovum. Thus it is said to be random or by chance encounter only. Next one is chemotaxis. In certain animals, sperms guide towards the ovum because the ovum releases certain chemical substances based on which it reaches the ovum directly. That is what we call it as chemotaxis. It is exemplified by certain organisms, fishes like sturgeon, Orthopixis caliculator, and also cyclostomes like lampreys. And the third type of approach of spermatozoon to the egg is sperm attractants. So several sperm attractants have been isolated from sea urchin. For example, one sperm attractant is called as resact, which is a small peptide found in the jelly envelope of the egg of Herbaceae punctuata, which is again an echinoderm. And based on these sperm attractants like resact, the spermatozoa approach towards the egg. And the second step is capacitation and sperm egg adhesion. This is advocated by a scientist called as F.R. Lilly by a theory called as fertilization theory. You have to remember this. F.R. Lilly's fertilization theory proposed in 1914. So what happens, what the scientist says, the change in spermatozoa which makes it capable of fertilizing the egg of the same species is known as capacitation. So the sperm is there, it has to fertilize. There are different types of okay. So there are different types of animals and the different types of ova are also present. And the sperm has to reach only the ova of the same species. So the mechanism by which are the changes that occur in the spermatozoa which make the sperm reach and fertilize only the egg of the same species is known as capacitation, so that the sperm comes in contact and adheres to the egg. And this is done by certain substances. According to F.R. Lilly, there are two substances involved. One is called as fertilizing, the other is called as anti-fertilizing. He says that fertilizing is present on the surface of the spermatozoa and anti-fertilizing is present on the surface of the egg membrane. Okay, and fertilizing is found to be a glycoprotein and anti-fertilizing is an acid protein. So the fertilizing of one species can bind with the anti-fertilizing of the same species but not of another species. Thus, coming together or union of gametes from different species to be is prohibited or it is reduced or it is minimized or it is not allowed because of a phenomenon called as capacitation, where the sperm with its fertilization comes in contact with the egg of the same species and adheres to the egg with the help of the anti-fertilizing. So in this diagram, you can see particle of fertilizer present on the surface of the egg which comes in contact with the particle of the egg, particle present in the egg membrane called as the anti-fertilizer. So thus there is a contact between these two which facilitates this. And the third one is said to be acrosome reaction and penetration of sperm. We know that the egg is said to be surrounded by a number of membranes. So before it reaches, and the main function is it to deliver its nucleus. So the sperm has to penetrate through the membranes, for which it is accomplished by acrosome reaction. So it can be studied in two headings. One is increased flagellar activity followed by acrosome reaction. In acrosome reaction, we see first of all exocytosis of acrosomal vesicle followed by extension of the acrosomal process. So let us discuss first about the increased flagellar activity. So there is a secondary egg membrane, that is jelly coat or primary egg membrane, whatever it may be. There is a direct contact with the egg membrane which increases the motility of the sperms, and this is accomplished by a decapeptide or a chemical substance released in case of sea urchins called as PRAC. Why I am mentioning only sea urchin but not the other organisms? In other organisms there are certain other mechanisms which are not yet revealed, and research may be still going on. So first of all, what has happens? The motility of the sperm is said to be increased when it comes in contact with the ovum. After that, the acrosome reaction takes place. So the first step here is exocytosis of acrosomal vesicle. So contact of the sperm with egg jelly, in case of sea urchins, it increases the localized fusion of the acrosomal membrane. So this is the acrosome. Localized fusion of the acrosomal membrane with the plasma membrane of the egg, so that the breakdown of acrosome takes place and it releases all the enzymes. It releases all the enzymes. These enzymes are called as lysins, which can dissolve the egg envelope in that particular area, that means in the local area, and clear the path for the sperm to reach the surface of the egg. And these lysins are different in different organisms. In case of mammals, as these are called as Jonah lysins. They pass through the primary egg membrane, that is Jonah pellucida. And in case of certain other organisms, we say it is a hyaluronidase, which dissolves the hyaluronic acid by which the cells of Jonah radiata of ovum are glued together. So this is the ovum around which there is Jonah radiata. So these cells are said to be joined by hyaluronic acid, a substance called as hyaluronic acid. Now hyaluronidase releases, and this hyaluronidase released removes all these hyaluronic acid, so that the Jonah radiata is dissolved and the sperm encounters the egg. So thus there is an encounter of the sperm with the egg. Now what happens is extension of the acrosomal process has to take place. So the membrane of the acrosome extends to form a thin tube called as acrosomal process, with which the sperm penetrates the egg. The head and middle piece of the sperm enter the egg, while the tail is said to be left outside. When you can observe this here in this diagram. So this black one is said to be, this is said to be the plasma membrane of the egg, and this constitutes the egg cytoplasm. So first of all, sperm comes in contact, and when you see the sperm, you see different parts. So this shaded portion is said to be the nucleus, and this dotted portion is the acrosome with all its contents, around which there is a sperm plasma membrane. Now what happens? Due to increased flagellar activity, sperm reaches the egg. That is the first thing. After thing, what happens here? exocytosis of acrosome vesicle. So the acrosome vesicle releases all its contents to outside. So this is the plasma membrane around which there is a primary egg membrane. Here we represent it as jelly coat. So all the acrosomal contents like jonalysins, hyaluronidase, commonly called as lysins, they are released and they dissolve the jelly coat so that the what happens now, the membrane of the acrosome, this membrane of the acrosome forms into a process, and this process is called as acrosomal process, which penetrates which penetrates through the egg membranes and reaches the plasma membrane, during which it penetrates with the help of the head and the middle piece. Up to middle piece means even the nucleus is exposed to the egg cytoplasm. Thus, the nucleus is delivered inside by the formation of an acrosomal process and contact of gametes. So, once the sperm penetrates the egg's outer membranes, the acrosomal process contacts the vitelline membrane of the egg. The egg is now released from its inactive stage and starts getting activated. Okay. At the same time, we have to also see another phenomenon called as prevention of polyspermy. So, if this is the egg, a number of sperms come in contact with it, but the egg has to allow only one nucleus, and other sperms have to be prevented from fertilization. This is accomplished. How? Let us see. First of all, fusion of sperm and egg plasma membrane takes place. Immediately after acrosomal process of sperm touches the surface of egg, the cytoplasm of egg bulges forward where the sperm has contacted. At the point of contact, it develops into what we call fertilization cone through which the sperm enters the egg. So where the sperm comes in contact with it, it forms a fertilization cone through which the sperm enters the egg. And the polyspermy, that means entry of multiple sperms, is prevented in two ways. One is called as fast block, the other is called as slow block. In fast block, what happens is some acrosomal proteins open the sodium channel in the egg membrane. So, this is the egg membrane and the sperm has penetrated inside, and now the sodium channel is opened and sodium influx takes place. So, lot of sodium ions enter. What happens when the sodium ions enter? We say that the membrane is depolarized. We have studied in the nerve impulse this concept. So, this depolarizes the egg membrane. So, the depolarized egg membrane will not allow further fusion of other spermatozoa. Thus, polyspermy is said to be prevented. And this process we call it as fast block. Why? Because it is extremely rapid. So, as soon as the first sperm enters, this process occurs and other sperm entry is said to be prevented. But though it is very fast and rapid, it is temporary process only. Depolarization may result again repolarization and it may come back to its normal state. Then polyspermy may occur. Then comes the process that is slow block to polyspermy. Okay. Slow block is initiated by fast block. Only when fast block is present, then slow block starts. Here, calcium ions either from stores of egg or from endoplasmic reticulum are released at the site of sperm entry, which initiate the cortical reaction. So, I have mentioned while dealing with the structure of the egg that Beneath the plasma membrane, there are certain vesicles of Golgi, and these are called as cortical granules because they are present in the ectoplasm, that is egg cortex. They are called as cortical granules. Now, what happens when the sperm comes in contact with the egg? Now, calcium channels are opened and calcium ions enter inside, which initiate cortical reaction. So, what is cortical reaction? Whatever these cortical granules are present, they come together and they form into a membrane around this sperm that has entered already. So, thus a membrane is formed after the sperm contacts the ovum, a process called as fertilization, and hence this is called as fertilization membrane. This fertilization membrane is formed by the fusion of the cortical granules, a process called as cortical reaction, so that the other sperms which are present on the surface of the sperm which haven't made an entry are further prevented by this cortical reaction, so that they cannot enter the cell egg at all. So thus the polyspermy is prevented by fast block, which causes depolarization due to sodium influx, and slow block, which is caused by calcium influx leading to cortical reaction, followed by the formation of fertilization membrane by the fusion of cortical granules, which prevents the polyspermy. So, if this takes place like this, the sperms come in contact with the egg. So, once one sperm enters the egg, now all the cortical granules release their contents and they form into a fertilization membrane around the what we call the penetrating sperm, so that other sperms are said to be prevented from their entry into the sperm. Thus, polyspermy is not allowed. Okay. Next, it is followed by metabolic activation of egg. I have already told you it is a secondary oocyte stage, that is, ovum is released, egg is released in case of mammals. Unless otherwise it is activated, second meiosis is completed, fertilization amphimixis is not possible. So, fertilized egg undergoes certain metabolic activities. First of all, increased rate of respiration. What do you mean by increased rate of respiration? That is nothing but oxidation of what we call the yolk content, other substances. For oxidation, during oxidation, what happens? Glycolysis, Krebs cycle, and electron transport, release of energy. So many things happen for which certain vitamins are required as coenzymes like activation of NAD and NADP. At the same time, increased rate of protein synthesis takes place, and this is what actually happens. Resumption of meiosis. That means the first meiotic block, first meiosis completed, in the second meiosis, in metaphase it is arrested. Now, this metaphasic arrest is prevented and it continues anaphase, telophase, cytokinesis, thereby the second polar body and the ovum is said to be released. And also scientists say that DNA synthesis is also started during the metabolic activation of egg. After that, what happens? The egg is now having the sperm nucleus and the egg nucleus. We call them as pronuclei because they are having only half the number of chromosomes. They are called as pronuclei. This is male pronucleus. This is female pronucleus. So both the nuclei fuse, a process called as amphimixis, resulting in the formation of a diploid zygote. Okay, so this is how the sperm comes in contact, and all the process like acrosomal license. Further, it releases the two things. One is male pronucleus it releases, and also proximal centriole. Remember that. And the male pronucleus fertilizes the female pronucleus, and the proximal centriole is responsible for what we call, it is responsible for the formation of first mitotic spindle. Okay, so thus a zygote is formed by the process of amphimixis, where the female pronucleus combines with the male pronucleus, forming a syncyrion or zygotic or diploid nucleus. And along with this, the proximal centriole is released, which forms the first mitotic spindle during cleavage. Okay, so this is what happens. This is the female pronucleus, and male sperm enters and releases the male pronucleus, and this proximal centriole is responsible for the formation of first mitotic spindle, so that the division starts, cleavage starts, and divides to form two cells. And coming to the significance of fertilization. Fertilization restores the specific parental diploid chromosome configuration by the fusion of haploid gametes of the parents. So, haploid gametes of the parents fuse to form a diploid zygote, which is having the same number of chromosomes as seen in their parents. Second one, it brings about genetic recombination as chromosomes of father and mother are in offspring, so that we acquire both the characteristics of the mother and father. Thus we show variations, we show a genetic recombination somewhat more specialized than either of the parents, because we are having both their characteristics. Thus we improve adaptation to ever changing environment due to this sexual reproduction process. Next, the third important thing is, in second meiotic block is said to be lifted off, and the secondary oocyte is said to be completing the second maturation division once the entrance of the sperm takes place and fertilization is completed. Next one, it causes rearrangement of ooplasm and organization into distinct organ forming areas. In the first, when I started this class, I've told you about something, predetermined fate of the certain organ forming areas of the cytoplasm of the egg, based on which the development can be mosaic or regulative. Thus here the ooplasm, it is organized into distinct organ forming areas. So this area forms, let us say, alimentary canal. This area forms, let us say, body wall, and this area forms nervous system, and all other visual organs from this area. Thus, here all the different areas of organ forming areas are said to be organized, rearranged in the ooplasm. Next, it introduces, the fertilization introduces proximal centriole in the egg, which forms the first mitotic spindle during first cleavage. It also activates metabolism of the egg, and genetic sex of the individual, that is XX or XY, is also determined at the time of fertilization. So these are the different um, what we call functions or aspects of significant aspects of fertilization. So fertilization results in zygote, which is single cell that has to undergo a number of mitotic divisions so that this multicellular body is formed. And all these mitotic divisions constitute the next process, that is cleavage. Are you there? You're following?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Coming to cleavage, it is defined as the progressive subdivisions of the zygote or an activated egg, we can call it in any way, by only mitotic cell divisions. No meiosis takes place here, by which there is increase in the number of cells as the number of cells is increasing, progressively the size is decreasing. So during cleavage, number of cells increases and the number of the size of the cells decreases. For example, we know that what is the largest cell? If anyone asks you, you say it is the ostrich egg. But at the same time, you say we cannot see a cell. A cell is microscopic, you say. But the cell which is called as largest, that is an ostrich egg, you can see with a naked eye. So this much is the large cell, but egg cell, fertilized egg, from which the cells of the body of the ostrich, which are microscopic, have to be produced. That is the reason why for each division, the cell which is naked, which can be seen with the naked eye, has to decrease in size so that ultimately it reaches the size which is that of the parental cell size. Thus, the divisions of cleavage are said to be can be synchronous or asynchronous. Once in synchronous mode, all the size gradually decreases. Once it reaches the size of the cell, after this, here there is no further decrease. With each division, similar sized cells are produced. Before what happened? The size is said to be gradually decreased. Once it reaches the normal cell, after that what happens? The size is said to be maintained. How this actually happens? We know between two mitotic divisions, one division followed by another division means there is G1 S G2 followed by mitosis. So one mitosis is completed and this is followed by G1S G2 and second mitosis starts. So for this to happen, it has to complete growth phase where actually this is the cell. If it divides to two cells and again divides immediately, the size is said to be reduced gradually. In case of normal cells of the body, somatic cells, the size should not decrease. The size has to be maintained, isn't it? So hence what happens? This cell, due to cytokinesis, which is equal, two half-sized cells are produced. Now they grow to the same size again, so that when they divide, they produce the same size cells. So this is how during G1 and G2 the size of the cell increases after one mitosis and before another mitosis starts. But during cleavage, what should be there? There is a progressive decreasing size. That means G1 and G2 are absent so that the cell continuously decreases in size. Let us see these characteristics one by one. What is the first characteristic? The unicellular zygote becomes a multicellular embryo. Second one is all the divisions are mitotic divisions only. As a result, the cells that are formed are called as blastomeres. So during cleavage, what are the cells that are formed? They are blastomeres. And here, these divisions are very rapid. Rapid in the sense that the interface is not absent. Interface is present, but only G1 and G2 are absent. They are absent. S phase is always present because this zygote is having 46 cells. It divides to form two cells which are having 46 each. That means without DNA duplication, 46 pairs of chromatids, if not are formed, here the daughter cells cannot have equal number of chromosomes. So S phase is present, G1 and G2 only are absent. Because G1 and G2 are absent, the rate of division is said to be at a rapid rate. If at all we say there is any growth during the cleavage process, scientists say this is insignificant. Next thing is the size of the cell decreases, but the size of the nucleus is maintained. That means karyokinesis here is always equal. And nucleocytoplasmic ratio. So this is the nucleus, this is the cytoplasm in the zygote. When it divides and forms these two cells, the nucleus size is said to be same, but the cytoplasm is decreased. Thus, the nucleocytoplasmic ratio gradually decreases during cleavage. As I said before, early cleavage divisions are synchronous, which become asynchronous during later stages. And coming to the speed or rate of cleavage, the rate of cleavage is determined by the cytoplasm rather than the nucleus. Why? This is the cell. After karyokinesis, two equal sized nuclei are produced. Now this is the term for time for cytokinesis. During cytokinesis, what happens? A constriction appears here which deepens so that both of them join and it divides into two daughter cells. Sorry, it divides into two daughter cells. So thus cytokinesis results in the production of two cells, isn't it? And for the cytokinesis to occur, it is not the nucleus, it is the cytoplasm that determines. Why they say so? Here, if the yolk is said to be present up to this point, up to the equator, yolk is present. So that active cytoplasm is present here, and there are two daughter nuclei formed on the other side in the active nucleoplasm place. Now what happens? Here, this is entire thing is said to be surrounded by, is said to be filled with yolk. So constriction cannot happen at this place because there is yolk. The actin filament cannot pull the plasma membrane where there is large amount of yolk. So the constriction moves to the other side where there is less amount of yolk, and here it takes place. And the karyokinesis is always at right angles to each other, this one, so that two daughter cells can be formed. So thus, what actually happens, what I want to mention is, it is the cytoplasm having the yolk that is responsible for enhancing the rate of cleavage. Okay, and here the distribution and the quantity of yolk influences the rate of cleavage per row, that is the constriction. And coming to the shape and size of the embryo, the shape of the embryo will not change. Why? Because we know that all these are happening within the egg membranes. So the egg membrane maintains the shape. The cells, though they increase, they will not change in the, they will not have any impact on the overall change in the shape and size of the embryo. Thus, there is no change in the total mass of embryo because the entire development takes place in the chick's egg only. Overall mass is not changing because with each division, the size gradually decreases. In addition to this, between the cells, new gap junctions and new tight junctions are said to be formed. And another aspect is metabolism. Metabolic activities occur at a rapid rate because lot of energy is required, hence metabolism has to take place, which involves breakdown of yolk, glycogen, etc. At the same time, for each division, S phase is said to be present between each successive division. So DNA synthesis continues, RNA synthesis continues, lipid synthesis also continues. And here, the spindle required for divisions also is formed simultaneously. In order to enhance the metabolism, oxygen consumption is also increasing greatly. These are the characteristics of cleavage. Now let us come to the planes of cleavage. The plane of cleavage is determined by first thing, that is the position of the mitotic spindle of the dividing egg, or we can also call it as after the egg divides it forms blastomeres. That means the position of the mitotic spindle of dividing egg or blastomere is responsible for determining the plane of cleavage. Okay. As a rule, the plane of cell division remains always at right angles to the long axis of the spindle. If the egg divides to form nuclei in this direction, the plane of division will be in this direction. If the nuclei are here in this direction, then what happens? The plane of cleavage will be in this direction. In case of amphibian eggs, which are said to be mesolithical, where active cytoplasm is present here, okay, and the daughter, the plane of cleavage cannot take place in this place because it is occupied by yolk. It is shifted somewhat above the equator. So the spindle, mitotic spindle of the egg and also the yolk determine what we call the plane of the cleavage. During cleavage, different cleavage furrows. These are nothing but constrictions. Divide the egg from different planes. Let's see the planes. The first one is a meridional plane. Meridional plane runs from animal pole to vegetal pole. That too from exactly through the center. So the cleavage furrow, when it passes through the center of animal vegetal axis and bisects or divides the egg into two equal sized daughter cells or blastomeres, it is called as meridional plane. So it is exemplified by cleavage furrows in Ranapipians, Lepidosteus, and first cleavage furrow of chick or meridional plane. Second one is vertical plane. The cleavage furrow passes through the animal pole to vegetal pole, but not through the center. That means if this is the egg, if it passes from the center exactly, it is meridional. If it passes either on one side of the median axis or on the other side of the center median axis, then it is called as a vertical plane. So vertical plane always results in two cells, one which is larger, the other is which is smaller. Exemplified by again Lepidosteius, osseous, frog, chick, and third cleavage furrow of another fish called as Emia calva and equatorial plane. So here we have seen the vertical plane which passes through the center or on either side, meridional and vertical. When we see the egg, there is another plane which is exactly between the animal pole and vegetal pole. We can call it as equator. So cleavage furrow that bisects the egg at right angles to the main axis. This is the main axis. At right angles means 90 degrees. So which bisects the egg at right angles to the main axis is called as equatorial plate. So even the equatorial division results in two equal sized daughter cells. Examples are first cleavage plane of eggs of higher mammals and fifth cleavage plane of Amblystoma maculatum, which is a salamander. Okay, and horizontal plane like vertical plane. If equatorial plane is said to be running through the center exactly, horizontal plane runs either above the equator or below the equator. So it is similar to the equational plane but goes through the cytoplasm on either side of it. It is exemplified by third cleavage plane of Amphioxus and frog, and as a result we come across with two cells. One is small, the other is large. Okay, and coming to the types of cleavage, there are different types of cleavage and these are based on the pattern of the cleavage, the potency of the egg, and also amount of yolk present in the egg. Let us see one by one quickly. Coming to the classification based on the pattern of cleavage, there are four types. radial cleavage, biradial cleavage, bilateral cleavage, and spiral cleavage. Why these different types? Because the organism's body is such that when you see the molluscal body, the body is spirally twisted inside the spirally twisted shell, which is highly coiled, as in case of pila. Okay, and as the body is organized, the cleavage is also organized. If the our body is said to be bilaterally symmetrical body, so bilateral symmetrical body has to be produced only when the cleavage is said to be bilaterally symmetrical. And coelentrates and poriferans exhibit a radial symmetry, the body organization, and the cleavage will also be radial pattern only. So animals differ in their cleavage pattern, that is in positioning and orientation of mitotic spindle differs in different organisms, based on which there are different types. The first one is radial pattern. In cleavage, radial cleavage, the each division, successive cleavage planes means is each division cut straight through the egg. The entire egg is said to be cut. So, if the entire egg is said to be cut, it forms two cells. If both the cells are cut, it forms four cells, and all the cells are equally cut, it forms 8 cell stage, and so on. In all these divisions, the entire egg is said to be cut straight through. And cleavage furrows are always perpendicular to each other, symmetrically disposed, symmetrically disposed around the animal vegetal polar axis. That is the reason why all the cells that are produced due to radial pattern of cleavage are all equal in size, hence they exhibit radial symmetry. We know what is radial symmetry. Here there is an onion. Onion has a number of what we call peels inside, which we consider which we can consider as its organs, and they are said to be not just oriented against one axis, but these peels are organized in many axes, so that when you cut through this axis, you can find two equal halves. When you cut through this axis, you can find two equal halves. In any way you can cut, you can have two equal halves. Likewise, even the blastomeres that are present here, 8 are present. When you cut it any direction, it produces two equal halves. Thus, due to radial pattern of cleavage, it produces an embryo with radial symmetry. In this type of, the first three divisions are meridional, and the third division is said to be horizontal. So, if this is the egg, take an onion, you cut it first, it forms two divisions. Second, it is said to be again cut in the at right angles to, this is the first cleavage division, which is, and second one is said to be at right angles to the first one, so that four cells are formed. This is not three. First two divisions are meridional, they run from the animal to vegetal axis through the center of the egg, and now we have four cells, isn't it? Now the second division is said to be horizontal. That means it cuts through the from the equatorial plate on either side, so that 8 cells are produced. 4 are said to be present upside, and 4 are said to be present below. If they are equally sized, all are blastomeres. If they are unequal sized, some are called as micromeres and the other are called as macromeres, which we are going to see in coming slides. And this is seen in echinoderms, poriferans, amphioxus, lampreys, lungfishes, and amphibians. And second one, this is how it looks like: 2 cell stage, 4 cell stage, 8 cell stage. All cells are in whatever the stage it is, all the cells are said to be equal in size. Even in the 16 cell stage, 32 cell stage, when you see, all the cells are said to be equal in size up to the formation of morula and blastula. And coming to biradial cleavage, it arises when the first three division planes do not stand at right angles to each other. We know biradial symmetry. Uh, uh, the example is a sea anemone. Okay, sea anemone has a cyclic structure with a mouth opening. So if this is the mouth opening, the organs are arranged around two axes. Some organs are arranged around this axis, and some organs are arranged around another axis, that is this axis. Okay, so if you cut the animal, if you cut the animal through this axis in any plane, the same axis in any plane, it can form two equal halves. So thus it forms one radial cleavage plane. If you cut the animal in this direction, because some organs are arranged around this axis, previously there are some organs are arranged around this axis. So all the organs that are arranged around this axis, they exhibit radial symmetry, and all the organs that are arranged this axis, when they are cut on different planes through the same axis, they also exhibit radial symmetry, and we call it as biradial symmetry. Here, the first three division planes are never at right angles to each other. In case of biradial cleavage, first two divisions are meridional, they're said to be at right angles to each other. But here, no, not even one continuous first three divisions are said to be right angle to each other. It results in biradial symmetry. And the third one is bilateral cleavage. The first cleavage plane passes through the animal vegetal axis and divides the asymmetrically distributed cytoplasm equally between the two blastomeres. Now, this cleavage plane corresponds to the future median plane. That means, what is our median plane? It runs through the center of the head till, if at all we have tail, like reptiles, if till it reaches the posterior part. So there is an axis which passes through the center of the head, anterior end to posterior end. This will be called it as central median axis, which runs antero-posteriorly. That is our median plane. And this plane which cleaves the egg in our embryonic development, during our embryonic development in the zygote, corresponds to our median plane. Thus, this becomes the right half and this becomes the left half of the body in the adult. And such a cleavage which distinguishes or bisects the um, egg into two halves, right half and left half, which correspond to the right and left halves of the body of the adult. It is called as bilateral cleavage. Okay, and it is exemplified in tunicata. Just it is not present in us. Just I am telling you to understand easily only. Ours is said to be highly evolved, and ours is said to be somewhat different. Whether it is blastulation, whether it is glastrulation, even cleavage pattern, everything is different in mammals. And bilateral cleavage is exemplified by tunicata. And coming to the spiral cleavage. So when you see the term oblique, you can identify that that question is related to spiral cleavage. So it results due to the oblique positions of mitotic spindles in the blastomeres. In first stage, these are said to be at right angles to each other so that it divides the cells into two. Okay, and in the second stage, in the cells, after karyogenesis, cytokinesis followed between the two nuclei. This is the plane. But here what happens is the first cell division results in two daughter cells, and the second division is said to be somewhat oblique so that the other two cells are placed not exactly one above the other. In case of radial symmetry, they are present one above the other. But in case of spiral cleavage, either they are present in left-handed direction obliquely or in the right-handed direction obliquely. and such a pattern is called as spiral cleavage. Where this actually is present? It is present in those where there is a rotational movement of cell parts around the egg axis. Thus, it can result in either sinistral or dextral orientation. And hence, when we study pila, that is snail, we see a shell which is dextral or sinistral. That dextral or sinistral shell is said to be based on the spiral cleavage. So exemplified by pila, that is mollusca, annelids, rotiferans, nematodes, and turbellarians. And finally, coming to the most advanced human being, most advanced mammals, where it is called as rotational cleavage. The name itself says rotational. That means one division takes place, it produces two cells. And second division in all the other organisms takes place simultaneously in both. But here, that is not the case. First division results in two cells. Next, the second division takes place in this. Okay? Thus, a T-shaped structure is formed. This is followed by the third division. Actually, this is the second division for this one, but for us it is third division, which takes place in this. Likewise, after that, it is shifted, it rotates, the division or the cleavage is rotated between the cells. Hence, we call it as rotational cleavage. The first cleavage is meridional, resulting in daughter cells. The second cleavage is rotational because the blastomeres divide in different planes. First blastopere divides meridionally, whereas the other blastopere divides equationally. So if this is dividing like this, the other one divides like this. So thus there are different configurations in the developing embryo as seen in mammals. Okay, so this is how it appears. The first division is meridional, resulting in two daughter cells. In the other one, it is meridional. In the other one, it is equational. Thus, as with each division, one cell dividing meridionally, the other dividing equationally, resulting in rotational cleavage. And in most of the examinations, this question is generally asked. Rotational cleavage is present in dash mammals, you can write it down. And coming to the based on potency. So based on potency, that means the developmental ability of the egg, it is of two types, determinate cleavage and indeterminate cleavage. In determinate cleavage, what happens is each blastomere has its characteristic position and unalterable fate. So if these are cells that are formed, okay, so if these cells, this group of cells are going to form body wall, they are going to form body wall only. If you remove these cells, the embryo during and after the entire embryonic development, it cannot form body wall at all. Thus, their fate cannot be changed. Their fate cannot be changed. It is unalterable. In that case, it is called as determinate cleavage. So, when the cleavage results in few cells called as blastomeres, which have a predetermined fate, such a cleavage is called as determinate cleavage, and it results in always mosaic development. Examples you can read through from this: ascaris, tunicates, mollusks, annelids, etc. Opposite to determinate cleavage, indeterminate cleavage. What I have said you before is zygote divides into two cells. In us, when these two cells are separated, each can form into one complete adult. Even if two cells divide to form four cells, if you separate four cells, each one can develop into one individual. Even at 8 cell stage, if you separate it into 8 cells, if it is able to develop into 8 different individuals, then we can say that no, even up to third cleavage, no cell fate is said to be determined. If at 16 cell stage, if you separate it into 16 cells, or you separate 2 cells from the other remaining 14 cells are intact and brain is not developed means these 2 cells have their fate determined at 16 cell stage. Thus, during this embryonic development, when the fate of the blastomeres is not determined up to some stage, not the entire embryonic development, up to certain stage, then it is called as a indeterminate cleavage. So up to certain stage, it is capable of developing into a whole embryo and finally giving rise to a whole adult. And such a development is called as, due to indeterminate cleavage, such a development is called as regulated development, and it is characteristics of vertebrates and echinoderms. Even as we have the indeterminate cleavage. That is the reason why we have uh, monozygotic twins. Why we can give rise to monozygotic twins? Because we have indeterminate cleavage. And coming finally to the cleavage pattern based on distribution of yolk. So already I told you, whatever the yolk that is present, it impedes the cleavage furrow. So cleavage furrow cannot be found here. It should be found in the region where there is active cytoplasm. So the distribution of yolk is also determines the cleavage, based on which the cleavage is of two types: holoblastic, meroblastic. Holo means complete, total. Blastic means the entire egg can be bisected from one pole to the other pole, either in meridional direction or in equatorial plane. Meroblastic means the successive cleavage divisions are restricted only to one part of the egg, like in the case of heavily telolesthal eggs. In heavily telolesthal eggs, this entire egg is said to be occupied by, what happened here? This entire egg is said to be occupied by yolk, leaving only this small space where the actual divisions takes place. That means the cleavage is said to be incomplete. Mero means half, partial. So only a part of the egg is said to be undergoing cleavage, hence it is called as meroblastic cleavage. Let's see one by one. Total or holoblastic cleavage. The cleavage furrow bisects the entire egg here. It is of two types: equal holoblastic cleavage, unequal holoblastic cleavage. In equal holoblastic cleavage, where it occurs in microlecithal or isolethal eggs, where the yolk is said to be uniformly distributed. So, first division results in two daughter cells which are equal in size. Second division, it bisects through the equational plane. It is meridional, it is equatorial, so that the resulting cells are all equal in size. When the division is holoblastic, equal. Example, Amphioxus marsupials, placental mammals, that means eutherians and metatherians and eutherians, like us. And the second one is unequal holoblastic cleavage. It occurs in mesolithial eggs or moderately telolithial eggs, like amphibians, where the yolk is present not below the equator, it is present up to the above the equator region. Large amount of yolk is present and the active cytoplasm is here. So the first division starts here from this direction and starts in the vegetal pole also in this direction, but rather it slowly passes, it comes fast, whatever it may be, it bisects the zygote into two cells. But the second division cannot start here because though it can pass through slowly, the other side is said to be initiated here and it is passing through and it is reaching the other side. But here, passing through the yolk at this portion in the equational plane is said to be very difficult. So it is the equatorial plate rather than equational plane. It is now shifted, that is horizontal plane where the cleavage takes place. Now it divides the cell into two. Thus, two cells which are smaller in size called as micromeres and two cells which are larger in size called as macromeres are produced. Thus, the cells that are produced are unequal. Hence, this is called as unequal holoblastic cleavage, exemplified by amphibians which exhibit mesolithal or moderately telolithal eggs. So, examples are lower fishes and amphibians. Coming to the meroblastic cleavage, which is also called as partial cleavage or incomplete cleavage. It occurs in only macrolecythal eggs or heavily telolecythal eggs or polylecythal eggs, and even in certain microlecythal eggs. It is said to be two types: discoidal cleavage and this is not macrolecythal eggs. Discoidal cleavage and superficial cleavage. Coming to the discoidal cleavage, it is characteristic of macrolecythal or heavily centrolecythal eggs. Here, cleavage is restricted to the disc-shaped active cytoplasm of animal pole. For example, if this is the egg, this is the animal pole, this is the vegetal pole, and yolk is present, it is filling this end. Sorry. So yolk is filling the entire vegetal pole. At the same time, almost it fills the animal pole also. So this entire thing is filled with yolk, large amounts of yolk in case of bird egg, prototherian egg, eggs of bony fishes, eggs of reptiles like turtles and elasmobranch fishes. So actual cytoplasm is restricted to this portion only. So all the divisions take place here only, resulting in what we call this area, which is almost seen from surface area as a disc-like thing, and hence the blastomeres appear after few divisions. They appear as a disc. Hence, this is called as a discoidal cleavage. And coming to the other one, that is superficial cleavage. This is found in only central acythal eggs. We know insect eggs contain yolk at the center, and the active cytoplasm is present towards the periphery where the nucleus is present. So all the divisions are restricted to the active cytoplasm only, and thus it is superficial. Cleavage is restricted to peripheral cytoplasm, hence it is called a superficial cleavage, exemplified by arthropods and insects. Okay, so these are the different patterns. Ultimately, it results in morulation followed by blastulation. In case of isolate eggs, it is holoblastic equal cleavage, resulting in all similar cells, equal size cells. In case of mesolithic eggs, like amphibians, holoblastic unequal cleavage occurs, resulting in micromeres and macromeres, unequal sized cells. In case of heavily telolithic eggs, it is discoidal cleavage, where all the divisions are restricted only to the animal pole, a disc-shaped area. And in case of central eggs, all the divisions are restricted only to the periphery, thus resulting in different types of blastula. So the blastula, the development of blastula, that is blastulation, in turn is depending on the pattern of cleavage. Let's see how it takes place. Before that, I would like to mention the laws of cleavage. There are four laws of cleavage: Sachs law, Hetwig's law, Balfour's law, and Pfluger's law. Sachs law states that cells tend to divide into equal size of daughter cells, and each plane of division tends to bisect the previous plane at right angles. So, if this is the first division and the second division is at right angles to the second division, the second division is right angles to the first division. Thus, the spheroidal shape of the blastomeres is maintained. That is what Sachs law says. Actually, based on these four laws only, these planes we have devised later. The second one is Hetwig's law. The axis of mitotic spindle occupies the longest axis of the protoplasm, and cleavage for row tends to cut it transversely. If mitotic spindle is like this, okay, then these chromosomes are separated to this side, and these chromosomes come to the other side. So after karyokinesis has taken place, what happens? The chromosomes come to this side in this direction. And now the cleavage for where it should start, it should not start here. Rather, it should start here. It cuts it transversely. Okay, the mitotic spindle occupies the longest axis of the protoplasm, but whereas the constriction, the cleavage furrow, cuts it transversely. The mitotic spindle is in this direction, so that the nucleus is formed on this direction, and now the cleavage furrow cuts it again transversely. So whatever it may be, according to him, the mitotic spindle occupies the longest axis of the protoplasm, and the cleavage furrow cuts it transversely. And Balfour's law is all about the rate of cleavage. So the rate or speed of cleavage is inversely proportional to the amount of yolk. That means if yolk increases, rate of cleavage decreases. If yolk decreases, as in case of microlysal eggs, it is very rapid, so that the cleavage rate is said to be increasing. And Fluger's law suggests that the mitotic spindle always elongates in the direction of least resistance. So mitotic spindle occurs in the direction of least resistance, means where there is no yolk, where there is active cytoplasm. That is what these four laws tell us about cleavage. Next, coming to the next part, that is mid-blastular transition. Do you have mobile data? Can I continue until blastulation is over? Should I stop
Continue,
here?
sir.
Okay. Because of mobile data, some have gone already. That's the reason I am asking. So next part is mid-blastular transition. Mid-blastular transition is a term coined by Newport and Krishna in 1982. What does it say? I've told you that during cleavage, G1 and G2 are absent, and all the divisions are synchronous. In later stages, it becomes asynchronous, isn't it? That means these G1 and G2 stages are added at a later stage. When the G1 and G2 are added, in S phase only DNA synthesis takes place. When G1 G2 are present, there is synthesis of RNA and synthesis of proteins, which continues even in G2. So the synthesis of RNA starts in blastomeres only at the end of cleavage, and these changes constitute mid-blastular transition. What actually happens during cleavage? As the initial part of the cleavage is very rapid, which should decrease the size of the cell, there is no growth phase. G1 and G2 are absent. When once the cell, when once the embryo reaches the uh, stage where the size of the cell corresponds to the size of the adult body, then G1 and G2 are added, so that one division is followed by G1 and G2, so it increases in size, divides, and the same size is to be formed. And this transition where G1 G2 is added and RNA synthesis starts, protein synthesis starts in blastomeres, is called as mid-blastular transition. And during this, what happens? All the maternal RNA are said to be cleared. Only the asynchronous cell division, which is programmed by the zygote. It is not directed by the mRNAs, heat shocked mRNAs, and the one masked mRNAs. They are not present now. All the mRNAs are said to be synthesized by the nucleus of the zygote, the chromosomes present in the nucleus of the zygote itself. These are programmed by the zygotic genome. And this transition is what we call it as mid-blastular transition is. So here, as a result of cleavage, it results in large, loosely arranged package of cells. So a number of cells are said to be formed, and these are loosely arranged inside the primary egg membranes. In each cell is having its own plasma membrane. These are present inside the primary egg membranes. And it is the multicellular embryonic stage with loosely arranged blastomeres formed as a result of cleavage. And this is called by scientists as morula, and the formation of morula as morulation. Why this is given? In Latin, morula means mulberry. And when the scientist saw this for the first time, he identified it with the shape of the mulberry fruit. For example, our pineapple fruit with all these small structures around it, we can compare the pineapple fruit with the mulberry fruit. For them, it is mulberry-like, so he called it as morula. And these cells are tightly packed within the fertilization membrane, and in morula, tissues are not yet formed. And morula is found to be absent in centrolithical eggs of insects. You have to remember these three things. The first thing is it is absent in centrolithical eggs. True morula is a solid ball of cells. True morula should be mulberry fruit like. So it must be a solid mass of balls, and that is said to be seen only in ciliates. Okay? Whereas in case of macrolithical and telolithical eggs, that means reptiles and birds, chick egg, where all the divisions are restricted to the animal pole, the morula is said to be in the form of disc of cells towards the animal pole. So these are the differences between the morula. It is not seen in central eggs. True morula with a ball, where it is in the form of a ball of cells, is seen in ciliates. And in case of macrolithical eggs or heavily telolithical eggs, it is present in the form of disc towards the animal pole. And from morula, the blastula is formed. The formation of blastula from morula is called as blastulation. So, as the cleavage proceeds, the number of blastomeres gradually increases, and they adhere to each other. They adhere to each other and form, first of all, a layer. They form into a layer, and this layer is called as the blastoderm, leaving a space inside called as blastocele. Okay, and this layer is said to be single cell thick in case of coelenterate, echinoderms, and amphioxus, whereas in case of vertebrates, the blastoderm is multi-layered. That is the main difference we have to observe. In case of coelenterate, echinoderms, and amphioxus, the blastoderm is single cell thick, uh, even some many cell thick, but in case of vertebrates, it is completely many cell thick layer, leaving a space inside called as blastocele. Let's see the morula, which is said to be ball-like with cells inside and outside, how it transformed to form the blastula, which is having space inside. There are two theories proposed here. One is called as hydrostatic pressure theory. Excuse me. The other is called as surface tension theory. Hydrostatic pressure theory is proposed by Dan. What he says is the cleaving cells release certain watery contents between the spaces present between the cells. So this watery content is what is responsible for generating hydrostatic pressure that pushes all the cells to the periphery, leaving a central fluids filled space, thus a hollow sphere of blastula, having a blastoderm means outside and a blastocele filled with fluid is formed inside. And another theory is said to be surface tension theory. It is proposed by Gustafsson and Wolfer. We know that on on a leaf, when you see a droplet, another droplet is also present here. You try to move the leaf so that when these two come together, they, both the droplets fuse to form a single droplet. And what is the phenomenon that is present here? It is the surface tension between the leaf and the cells that is responsible for coming together of these cells. Likewise, according to Gustafsson Walpert, what they say is the cells during the cleavage, as they are producing cells, the cells are said to be rounded like the droplets. And these rounded cells exhibit what we call surface tension, which makes them coming together, forming into a layer, leaving a space at the center according to surface tension theory. And coming to the blastulation process, formation of blastula, and this blastulation process is affected by the yolk content in different types of eggs. So we start with isoleucithal eggs. In isoleucithal eggs, what happens? All the divisions are said to be resulting in equal sized daughter cells, so that all the cells are said to be equal in size with an outer blastoderm and inner blastocele, as exemplified by echinoderms, mollusks, and ascidians, urochordates. The exception is there is an exception with respect to the isoleucithal eggs of chordates, exemplified by amphioxus. Here, the cells towards vegetal pole are somewhat larger, not big. We cannot distinguish them as micro and macromeres, but when compared with the cells towards the animal pole, the cells towards the vegetal pole are somewhat comparatively larger in case of chordates, exemplified by amphioxus. But in general, all the cells are said to be same in the blastoderm, which arises from the blastula, which arises from isoleucithal eggs. And coming to blastulation moderately teleucithal eggs. Moderately teleucithal eggs are exemplified by amphibians. We know that. Here, the first division is said to be at right angles. Second division is at right angles to the first one, and the third division is somewhat above the equator, resulting in micromeres and macromeres. And the blastoderm is said to be also having micromeres towards the animal pole and macromeres towards the vegetal pole. This forms the floor and this forms the roof. And such a blastula is seen in moderately teleucithal eggs. Coming to this is heavily. Blastulation in heavily teleucithal eggs like fishes, reptiles, and birds, where the blastulation is restricted only to the germinal disc at the animal pole. It is restricted only to this place. So, morula is disc-like and blastula is also disc-like. And here what happens actually is, listen carefully, I will take this space. So, this is the egg with yolk. So, this entire thing is filled with yolk. Okay. Now, all the divisions are restricted to the animal pole only. So, it forms into disc-like structure. And we have already seen that in all the vertebrates, it is the blastoderm is multicellular. It is thick multicellular. Okay. So, at first, both are said to be connected. The cells are said to be overlying the yolk. There is no space between. After that, what happens? The cells here, they shift towards animal pole. They are lifted towards animal pole, leaving a space between. Okay, they leave a space between, and hence what happens here is a space is created between the outer blastoderm and yolk, and this space is called as subgerminal cavity or segmentation cavity. This is called as subgerminal cavity or segmentation cavity. And when you see from the above, when you see from the above, the central portion, this portion, because there is a cavity here, it seems somewhat transparent. It seems somewhat transparent. And this central portion is called as area pellucida. Whereas the peripheral portion surrounding at the center, this is the center, and towards the periphery, the entire periphery appears to be somewhat opaque because below there is yolk content, and that peripheral portion is called as area opaca. Okay. This is what here they are telling us. Blastulation is restricted to the germinal disc at the animal pole. The cells in the central part of the germinal disc are multilayered and are separated from the underlying yolk. These cells are said to be separated from the underlying yolk, while the marginal cells are in contact with the yolk mass. So the marginal cells are in contact with the yolk mass. This forms a central clear portion called as area pellucida because there is segmentation cavity and yolk is said to be not there, and the peripheral dense portion which is opaque is called as area opaca. So area pellucida is transparent central portion and peripheral portion is called as area opaca because it is opaque dense. Next, as the central cells separate from the yolk, subgerminal cavity appears. So we have said this is the subgerminal cavity that is appearing. The outer layer of cells forming the roof of subgerminal cavity is called now as epiblast. So now this layer, this entire layer, blue colored one is called as epiblast. Next, a process called as delamination or involution or infiltration. Don't ask me about this. Tomorrow, in the tomorrow's morphogenetic movements by gas during gastrulation class, I'm going to address them. So just remember this. Either by a process called as delamination or involution or infiltration, which is also called as polyimmigration, cells of the lower surface of epiblast, that means these are the epiblastic cells, they are multi-layered. Below there is this entire thing is yolk and this is subgerminal cavity. So these are the lower surface cells which are present just above the subgerminal cavity. So these are separated from the above cells to form the primary hypoblast. So that's what happens now. There is epiblast here followed by primary hypoblast here followed by subgerminal cavity and yolk. So this is yolk, this is subgerminal cavity, this is primary epiblast, and this is epiblast. And a new cavity is formed between epiblast and hypoblast because this is epiblast with three layers. Let us say first two layers are separated from the third layer. So between these two there is a space created, and that space is now called as blastocele. So the primary hypoblast is separated from the epiblast by a blastocele. Now let us see this in the form of a diagram. So this is the epiblast and this is a subgerminal cavity or segmentation cavity. This entire thing is yolk. When you see from above, because it is separated from this, you see the central portion as transparent called as area pellucida, and towards the periphery the cells are in contact with the yolk, hence it is area opaca. And now what happens? Cells by ingression, that means cells from the margins they go inside, okay, or delamination. Just now we have spoken. The first two layers are separated and one layer comes down. So a new layer of cells is formed, and this new layer is called as primary hypoblast. Between epiblast and hypoblast there is blastocele, and between primary hypoblast and yolk there is subgerminal cavity. And this entire blastula is called as discoblastula. So let us see, we have seen the blastulation process which results in different types of these blastulae. There are six types of blastulae. One is coeloblastula, not colilo, it is coeloblastula, stereoblastula, periblastula, discoblastula, amphiblastula, and blastocyst. So within 5 minutes we are going to close it. Coming to coeloblastula, sorry, it's again coeloblastula. It is seen in Amphiochus and Echinoderms. The name itself says the blastula is with a coelom. Here coelom corresponds to cavity. That cavity is nothing but blastocele. So when the blastocele is surrounded by a single layered cells, that is blastoderm, which is also called as ectoderm by later scientists, this is called as coeloblastula. Okay, blastula is in the form of a hollow sphere with outer single layered blastoderm and inner blastocyst filled with mucopolysaccharides, that is coeloblastula. Coming to the stereoblastula. Stereoblastula is seen in spirally cleaving cells. It is seen in spirally cleaving cells where, because of these divisions, the blastocele is absent. There is a blastoderm with cells which are micromeres and cells towards vegetal pole macromeres, and there is no blastocele because it is already filled with cells. So it appears like a solid ball of cells. It appears like a solid ball of cells, like morellites we can see. But the difference between morella and this one is, in morella there is no layer. All the cells are randomly placed. But in stereoblastula, the outer cells are said to be formed into a layer, and even at the center is said to be completely filled. Hence, it is like a solid blastula. The solid blastula is called as stereoblastula, as seen in Annelida, mollusca, planarians, and Dimartinians. And periblastula. It is seen in insects. So, insects there is a central acephalate with central cytoplasm. So, whatever the blastula that is formed by the divisions that occur towards the periphery, and hence we call this as a periphery blastula, and this is also called as superficial blastula. Here also blastocele is absent as it is filled with yolk material. Thus, blastocele is absent in one stereoblastula. It is absent in two, that is periblastula. You have to remember these two things. Coming to the last three ones, discoblastula. It is seen in heavily telolestal eggs, fishes, reptiles, and birds, where there is discoidal cleavage. It is in the form of multi-layered flat disc separated from the underlying yolk by a segmentation cavity or subgerminal cavity, which we have discussed just now. And the next one is amphiblastula. Amphiblastula is the one that is present in cycon. So it shows holoblastic unequal cleavage resulting in micromeres and macromeres in the blastula stage. But these micromeres show flagellae towards the inside. After this, what happens is, towards the vegetal pole, the macromeres show a large opening. Okay. Now what happens is, the micromeres pull through the hole and they get what we call a process called as inversion, that is not seen in any animal throughout the animal kingdom. So the end of this, what happens is, there are micromeres on one side and macromeres on the other side. This both is eventually filled and followed by the flagella coming out. Thus, we can see two types of cells. Cells which are said to be flagellated micromeres and non-flagellated macromeres. Flagellated micromeres are present towards the animal pole. This is the animal pole. Even after inversion, this is the animal pole only. And non-flagellated micromeres are present towards the vegetal pole. Thus, there are two types of cells. Amphi means both, means two. As there are two types of cells, this blastula is given the name amphiblastula. And coming finally to the blastocyst. This is seen in mammals. So mammals have microlisthal legs. So microlisthal legs means we should exhibit radial cleavage, and we should have radial symmetry, isn't it? But we are having bilateral symmetry, and we are having rotational cleavage. So it results in this structure. The blastula here is called as blastocyst. The irregular cleavage, the rotational cleavage, results in a small blastocele which becomes progressively larger. There are two types of cells here. The first is said to be an epithelium. The first is said to be an epithelium. So this epithelium is said to be called as trophoblast. Scientists call this as trophoblast. And there is another group of cells which hang from the epithelium. And this group of cells is called as inner cell mass. And they are displaced towards only one pole. Let us say this is the animal pole. So towards the vegetal pole, there is no inner cell mass. And here there is a cavity, that is blastocyst. So here such a blastula which is in the form of cyst. Cyst means a cavity surrounded by layer with few cells, and it is an as if it is a cyst-like thing. Hence, this blastula is called as a blastocyst. That is characteristic of mammals. So these are the different types of blastulation techniques and different types of blastula in different organisms. In tomorrow class, we'll discuss about fate maps and gastrulation. Before that, let me close with this. Significance of cleavage and blastulation. These two processes are responsible for the formation of presumptive organ forming areas of the future embryonic body, so that they are segregated into definite parts of the blastoderm. If this is the blastoderm, this blastoderm is having all the cells. In the first class, we have discussed about triploblastic animals. That means some of the cells are destined to form endoderm. That means these cells form into endoderm. Some of the cells are destined to form mesoderm. These cells form into mesoderm. And the remaining cells form ectoderm. Okay. And even in the ectodermal cells, certain cells form only body wall, certain cells form only the nervous system. That means these areas of the blastula are produced due to different cleavage patterns, where the organ forming areas of the adult or the embryo are said to be localized in their respective locations. We call them as presumptive. That means these are the first organ forming areas from which the future organ forming areas are said to be formed. And cleavage and blastulation permits the migration of these cells. One, for the formation of the major presumptive major forming areas, and the second one is migration of the cells. What happens after blastulation? Gastrulation takes place, where the cells that are destined to form endoderm, they migrate inside. After this, what happens? The cells which are destined to form coelom, they migrate inside. Thus, an outer ectoderm, basal mesoderm, and inner endoderm is said to be formed. Thus, the blastocyst permits the migration and rearrangement of these areas during gastrulation. That is what the importance of cleavage and blastulation is. So, we'll meet tomorrow for fate maps and the final topic, that is gastrulation. Hope you understood. If any doubts, leave a message in the group. I'll answer. Okay.